Hello, I'm Tanwi Biswas from IBM Research, Tokyo, Japan. I will be presenting our research on effective in-silico gene perturbation by machine learning model interpretation for immunotherapies. T cells are the most powerful immune cells of human body which recognize and eliminate infectious and cancerous cells. For treating cancer patients in a more effective way with immunotherapies such as tumor infiltrating lymphocyte and chimeric antigen receptor or CAR T cell therapy, it is very important to understand the functionality of T cells. Recent advancement in the single cell profiling technology has enabled large scale accumulation of single cell RNA sequence data, and modern machine learning technology can provide new approaches to predict complicated functionalities of T cells. The gene PRF1 encodes perforin, which is highly relevant to immune T cells, contributing actively to cytolysis. This study aims to identify key transcription factors responsible for regulating the PRF1 expression in the CD8 T cells, leveraging machine learning model interpretation technique to perform transcription factor perturbations for contributing to engineering T cells. In this study, we have utilized a publicly available tumor infiltrating lymphocyte datasets. Before applying it to train the machine learning model, the dataset has been pre-processed following several steps such as quality control, batch effect removal, and log normalization. The workflow of our proposed method consists three major steps. In the first step, a gradient boosting machine learning model has been trained to perfectly predict the expression level of PRF1 from the given transcription factors as input. In the second step, the key transcription factors regulating the PRF1 expressions has been obtained leveraging machine learning model explanation techniques such as SHAP, which provides cell-by-cell gene attributes describing the contributions of the input transcription factors for predicting the target gene. Then, clustering of these sub-values has been performed. Our final step involves transcription factor perturbation, where we have intentionally changed the value of key transcription factors to its maximum or minimum and have observed the model output as perturbation. By comparing the average PRF1 value of the original test data and the model predicted value, it can be observed that the trained machine learning model can predict PRF1 value from the transcription factors successfully. The figure representing the clustering of the sub-values imply that different groups of CD8 T cells are being regulated by different transcription factors. Sharp summary result for each cluster provide transcription factors controlling that specific group, delineating finer cell mechanism. Due to lack of enough space, here we have presented top 5 transcription factors considering the whole CD8 T cells, and their perturbation effect has been summarized in this table. From the perturbation result, we can observe that PRF1 value has been increased while increasing the values of JEB2 and BH-LH-E40 to maximum, and decreased PRF1 value has been obtained for decreasing the values of JEB2 and BH-LH-E40 to minimum. On the other hand, decreased PRF1 value has been obtained for increasing the values of FOS, JUNB, and TCF7, and vice versa. And this perturbation result is perfectly consistent with the sharp summary result. In conclusion, this study focuses on identifying key transcription factors for regulating PRF1 expression in the CD8 T cells. The proposed method successfully identifies key transcription factors responsible for predicting PRF1 values. Our simulated perturbation experiment has provided promising result. The maximum increment of PRF1 value has been obtained for JEB2, which is about 19% from the originally predicted value, and maximum reduction, which is about 17% of the predicted PRF1 value, has been obtained for TCF7. We hope this study will provide new insight to physicians and biologists for conducting relevant research on drug design, gene knocking out experiments, and so on. Thank you so much for listening.